Hello, I'd like to thank the organizers for this chance to speak and all of you for listening. A key theme of the synthesis era going back to Fisher was the rejection of non-Darwinian theories held to be incompatible with genetics or population genetics, including the idea that evolution might exhibit tendencies or trends due to internal tendencies of variation. For instance, when Simpson integrated paleontology with this neo-Darwinian view, he looked at apparent trends in the fossil record. He argued that they probably were not real trends, and he concluded that even if the trends were real, they must be due to selection because selection is the only known cause of direction. The only alternative would be vital urges or cosmic goals without known mechanism. Now, where did Simpson get the confidence to reject an entire class of theories like this? Why does he say there's no mechanism when previous thinkers had simply assumed that biases and variation would cause evolutionary biases? We know the answers to these questions. Simpson and the architects of the modern synthesis did not reject the possibility of biases and variation. They rejected the notion that such biases could be effectual, that they could influence the outcome of evolution, an idea that they thought was incompatible with population genetics. And we know this because Haldane and Fisher made this argument explicitly, that because mutation rates are small, pressure is a weak pressure, unable to overcome the opposing pressure of selection. It can only be important if mutation rates are abnormally high or selection is absent. And this argument was cited and repeated by the architects of the modern synthesis and played an important role historically. But more importantly for our purposes, this argument represented the state of the art through the 20th century on the issue of internal biases and variation. So for instance, in the literature of molecular evolution, we still see the reasoning of Haldane that associates mutational effects with neutral evolution. That is, mutational explanations for patterns are assumed to rely on neutral evolution. And it's not just molecular evolution. So here, Stephen Jay Gould, in his giant book on the structure of evolutionary theory, he cites Fisher's argument about mutation pressure, and he concludes that since orthogenesis can only operate when mutation pressure becomes high enough to act as an agent of evolutionary change, empirical data on low mutation rates sound the death knell of internalism. And this famous 1985 paper that really defined the issue of developmental constraints cites the Haldane-Fisher argument as a barrier to recognizing the efficacy of developmental biases and variation. The authors suggest that this Haldane-Fisher argument deserves re-examination here, but they did not offer a re-examination, and they were criticized for this, for not providing a mechanism. And in evo-devo today, it's widely believed that population genetics cannot provide a full account of the influence of development in evolution. Because this way of understanding the role of mutation as a pressure has been influential, it's important to understand why it's misleading. An evolutionary process that depends on events of mutation that introduce new alleles will be subject to biases in the introduction process by a simple first-come, first-served dynamic that Haldane and Fisher did not consider. This was first demonstrated in 2001 using simulations with a simple model of one-step adaptation with two choices. So imagine all these different possibilities here. There are two of them that are beneficial. One of them, this green one going to the left, is not as high. The selection coefficient isn't as large, but there's a larger mutation rate. And then this other one has a larger selection coefficient, but a smaller mutation rate. And the question is, how often will evolution go toward the mutationally favored option? And here are some results of simulations. And here the bias and outcomes going to the left versus going to the right is shown as a function of mutation bias for a bunch of different population sizes that represent low mutation supply to high mutation supply. And in every case, the lines are going up, and whenever the lines are greater than 1, that means that evolution is favoring this mutationally favored option, even though it doesn't provide the highest fitness benefit. Haldane and Fisher did not address this kind of effect because their conception of evolution and evolutionary causation effectively assumes evolution from standing variation. If the alleles relevant to the outcome of evolution are present initially, then mutation does not introduce anything new, but can only shift the frequencies of alleles that are already present, just like selection and drift shift frequencies of alleles that are already present. But the shifts for mutation are trivial because the rate of mutation is so tiny. And we can model this view in the same model. All we do is just take this model, but put the alternative alleles into the initial population, and this is what happens. So this is a case of this is a conditions of new mutation here, like I showed you before. But these bottom two curves here are when the alternative alleles are present in the initial population, even at low frequencies of 0.5% or 1%. There's basically no effective mutation bias. Now, mutation is still biased, but mutation bias doesn't matter unless it's biasing new alleles being introduced. So this is one reason why I'm always referring to the introduction process, or biases in the introduction process or origination process, rather than just repeating the words mutation bias. Whether a mutation bias will cause an introduction bias is a matter of population genetics. The second reason to emphasize the introduction process is that mutation biases are not the only possible source of introduction bias. Even if all mutation rates are the same, we could still have developmental biases that reflect asymmetries in a genotype-phenotype map. Okay, so that was the initial theory on this kind of effect, and so far there are 5 different research groups that have contributed to our theoretical understanding of this issue. So recently, Suarez did a kind of full analytical treatment of this one-step model with 2 choices. There's a really interesting paper that goes over a wide range of conditions with this model of 2 competing traits. Kano and colleagues did a lot of simulations of a codon-based model, and they formulated this really interesting way of capturing the total influence of mutation in a single coefficient of influence called beta, which can go from 0, no influence, to 1, a proportional influence. And then there's several groups that have modeled adaptive walks on complex landscapes. And in particular, this work from Ard Lewis's lab provides a more fully worked out theory for why evolution prefers certain intrinsically likely forms, which is one aspect of what Stuart Kauffman meant by self-organization. So some RNA folds are widely distributed in sequence space, just like some phenotypes are widely distributed in genotype space. And because of this, these frequent forms have more mutational arrows pointed at them from other regions of sequence space, and this makes them more findable by an evolutionary process that explores new possibilities by mutation. So what these theoretical modeling tells us is a number of things are important, like mutation supply, the diversity of the target. There's a number of outcomes of interest that have been shown, like this coefficient of mutational influence, systematic trends, the findability of common structures. And in general, what this theory does is it allows us to it gives us a warrant to draw this green arrow of causation that connects cause and effect. We have a way of connecting this. And in a broader context, this means that there's a possible causal link that exists between mutational and developmental causes of tendencies of variation and interesting evolutionary patterns. So we have a rigorous basis to pursue hypotheses of this form. And we now have plenty of evidence for this kind of causation. So the mutation pressure theory says that the influence of mutation depends on high mutation rates or neutral evolution, but this is not required for the efficacy of biases in the introduction process. So if we see ordinary mutation biases influencing the changes involved in adaptation, not requiring neutrality, not requiring high mutation rates, that's evidence against the pressure theory and for the theory of biases in the introduction process. So that's where we're looking for evidence of these mutational effects on adaptation in the lab or in nature. And the lab studies, of course, are the best for showing a causation in a direct way. So here are two studies in particular. So on the left, we see the results of the evolution of rifampicin resistance, showing a strong correlation between the frequency with which a resistant variant evolves in replica cultures and the rate of mutation to that variant. That's from McLean and colleagues. And on the right, we have data on about 90 cefotaxime-resistant isolates from 3 different parental strains that have different mutation spectra. So resistant strains in blue here come from mut-T parents, and mut-T favors AT to CG transversions, which are grouped together in this box. That's why most of the strains in this box are the blue ones. The red ones are resistant strains from mut-H parents, and they tend to have the transitions that are favored by mut-H. And then there's wild type strains from a wild type parent as well that have multiple types of mutations. So what this study shows is that you shift the mutation spectrum, you shift the spectrum of adaptive changes. That's a direct causal link. From a couple of studies here, again, from the laboratory. What about adaptation in nature, in which adaptive changes have been traced to the molecular level. So in recent years, sufficient data have accumulated to test for mutation bias among some of these famous cases of phenotypic adaptation traced to the molecular level, to tetrodotoxin resistance, for instance, adaptive changes to hemoglobin in high-altitude bird populations. This is representing resistance to cardiac glycosides and so on. And if we take a dozen cases like this in diverse taxa affecting diverse proteins, we see an excess of transition. So these are all cases of parallel evolution. So in each case, the number of events is at least 2. So for instance, in this second row here, tetrodotoxin resistance can be achieved or has been achieved in parallel by 8 different types of amino acid changes. All right, 3 of them are transitions and 5 are transversions, and the 3 transitions have happened 2, 6, and 3 times, and the 5 transversions have happened 2, 2, 2, 3, and 3 times. That's how to read this data here. And when we sum up, what we see is there's about a 1-to-1 or 1.3-to-1 ratio of transitions to transversions instead of a 1-to-2 ratio. So there's basically a 2-fold or 2.5-fold bias toward transitions relative to a null expectation where mutation bias has no effect. At an even broader level, we have evidence for mutational effects on innovation, particularly in regard to RNA folds. So as I mentioned previously, the theory says that RNA folds that are widely distributed in sequence space are more findable in evolution. So on the right here, we have a plot of the top 40 most common RNA folds in sequence space. And this is their frequency in sequence space as a function of rank, and the ones that are circled in yellow are the ones that are found in nature. So in nature we find the most common folds, and this is evidence for this findability effect, which Ard Lewis's lab calls the arrival of the frequent. So to summarize, the evidence comes from the lab and from nature. It addresses efficacy as well as scope or importance, and it covers what we usually think of as microevolution within a population and macroevolution over millions of years. And as you'll notice, most of this work is very recent, just like most of the theoretical work was very recent. And one of the things that means is that we're still figuring out new things. We're still surprised sometimes by the implications of this. And I'm going to tell you about one of those little surprises. It came as a surprise to me and a bit of an embarrassment, indeed, in regard to transitions and transversions and the conservative transitions hypothesis. So in protein sequence evolution, changes by transitions predominate over transversions. And this might be due to mutation bias, but it's often been suggested that transitions are more conservative at the amino acid, at the protein level. To test this requires a lot of fitness measurements, which were not available until recently. But now that we have them, we can just take a large number of measurements from deep mutational scanning studies, and that's what we've done here. This is about 11,000 different fitness measurements of single nucleotide changes causing amino acid changes in proteins, and we've converted those to fitness quantiles and aggregated them. And as you can see, there's some subtle differences between the 12 different nucleotide mutations, but if we aggregate them into transitions and transversions, there's hardly any difference. So considering the whole distribution of fitness effects, transitions just aren't more conservative than transversions. And we also looked at some studies of adaptation to see what was going on at the very high end of the distribution of fitness effects. And we used rank order statistics for this. So if you ask, what's the chance that a randomly chosen transition will be more fit than a randomly chosen transversion, the null expectation is 0.5, because just if 2 things are drawn from the same distribution, then the chance that this will be greater than this is just 50%. So we asked that for these 5 different adaptation experiments. And what we found, and this is called AUC here, this chance that a transition is more beneficial than a transversion was actually less than 50% in every case. And in the combined data, it's kind of on the margin of statistical significance here. And so when I saw this, I just laughed and I said, haha, isn't that ironic? Transitions aren't more conservative than transversions, and in fact, the beneficial transversions are more beneficial. But when I thought about this some more, I realized that this inference is not safe, and it may be wrong, because the membership in this set of adaptive changes, the adaptive changes that come to our attention, are conditioned on their degree of benefit and on their degree of mutational likelihood. And that kind of joint condition can induce non-causal associations and even statistical paradoxes like Bergson's paradox. So let me just explain what that means. So let's consider a simple example and suppose that achieving celebrity status is based on a combination of talent and looks. And talent and looks, let's say, are not correlated in the starting population. So we just get this symmetric cloud like this. But if celebrities are only the top 5% of talent and looks, then they are represented by this green subset here. And within this green subset, there's clearly a negative correlation between talent and looks, because an individual who achieves celebrity status with only modest looks was very talented, and likewise, one who achieves celebrity status with only modest talent was very, very good looking. So we could be seeing the same thing for mutation and selection, like in the case of transitions and transversions. It might be that the transversions that come to our attention, in spite of having a slightly lower rate of mutation, have a slightly higher fitness benefit to make up for that. And here's a suggested example from a recent study of clonal haematopoesis by Watson and Blundell. So I just took this just appeared in bioRxiv like 5 weeks ago, and I took this figure and I replotted the data here to show this negative correlation. So as we age, the composition of our blood changes due to a mutation selection process among the blood precursor cells, some of which overpropagate. In this some kind of somatic overgrowth, it's like cancer except benign, and it changes your blood composition. So what's shown here is a substantial negative correlation between the inferred mutation rate and the selection intensity for these drivers of clonal haematopoesis, suggesting that the drivers that come to our attention in spite of relatively small mutation rates tend to have higher growth advantages and vice versa. But this is the posterior distribution after mutation and selection, and we don't know what the prior distribution looks like. So we can't really be sure that this is a negative. I mean, it's a negative correlation, but we can't be sure it's a non-causal association induced by conditioning. So how can we approach this problem in a more systematic way? What do we have to know? What should we be looking for? So in general, we're looking for some In general, we're interested in something about a joint distribution of mutation rates and selection coefficients, and this joint distribution changes from a prior distribution, a distribution of mutations, to a distribution of mutations to a distribution of evolved changes. Okay, so selection samples from the actual, but mutation samples from the possible. So we begin with what I'm going to call a nominal distribution, defined for some abstract set of possibilities like all single residue changes. And for each change, there's a mutation rate and there's a selection coefficient, and we can plot out the joint distribution here. Multiplying by mutation rate then gives the de novo distribution. So this is what mutation looks like. So when we multiply mutation by mutation rate, we're just shifting the density to the right. So these dots over here get fainter, and the ones to the right get darker. That's the effect of mutation on the distribution. And then when we go from the de novo mutation distribution to the fixed distribution or the selected distribution, then that's going to shift the density up along the y-axis here. So what kinds of correlations do we expect to emerge? We've approached this issue mathematically, starting with toy models with just a few mutation rates and selection coefficients. And here's some of the results here. I'm not going to go into the detail here, but it's complicated. We can see different kinds of effects, different kinds of associations. We can see the strong Bergson's association, where you go from a non-negative joint distribution to a negative one, and what we call weak Bergson's, which is just more negative than what it was before. But we can also get positive associations. And it really depends on what's the shape of the nominal distribution. So what we really need to do is to look at some actual data. So the distribution that I showed in the previous slide is actually from real data. So these top three panels are the ones that I showed before. This is based on a high-throughput survey of mutation rates and fitnesses of mutants in dengue virus from the lab of Patrick Dolan that they just published last year. So the nominal distribution on the left has 325 beneficial changes that are observed when dengue is grown on human cultured cells. So in this joint distribution, there's a slightly negative correlation of 0.06. In the de novo, where we get to get the de novo, we just multiply by mutation rates. And this is not a complete, we don't have, so we have measured values for the selection coefficients, but for mutation, we just have a model. So we multiply through by those mutation rate classes, and we get a de novo distribution. And then here, we're actually running a simulation of mutation and selection to get the fixed distribution. And there's 3 different versions of this shown here for different degrees of mutation supply. So at the lowest mutation supply down here, we're getting quite a strong negative correlation. But at higher mutation supply, it's not quite as negative. So that's one example. Of course, we'd like to find a case where we have both the nominal distribution and the fixed distribution without using simulations. And we can get that from cancer data, although the data are a little bit sketchy for this purpose. So I'm going to talk about, just briefly show you some results from P53. So for the nominal distribution, we have fitnesses from deep mutational scanning. It's important to remember that P53 is the guardian of the genome, and cancer driver mutations break it. So there's a lot of beneficial mutations, like 1/4 of them are beneficial. The mutation model is just the standard cancer mutational signature model that's derived from pan-cancer data from the Cancer Genome Atlas. And then for the fixed distribution, we're drawing on data on the prevalence of different drivers from the Gini database. And what we get is shown here. So in the nominal, there's a slightly negative correlation. In the de novo, which we get just by multiplying through the mutation rate model, shifting everything to the right, shifting the density to the right, there's a tiny, tiny positive correlation, 0.01. And then in the fixed distribution from cancer prevalence, there's actually a positive correlation. And when we do simulations of this, what we find is that we get the highest correlation between observed and simulated results at sort of moderate mutation supply here. And that's also where we get the highest correlation between, the most positive correlation between mutation rates and selection coefficients. So we know that mutation supply is a factor here in the shapes of these correlations. Now I'm going to show you one more result. So these are for single nucleotide mutations. Because the mutational signature model we have for mutation rates applies to single nucleotide mutations. But there's also doubles and triples. So there's 380 possible types of amino acid replacements. Only 150 of them happen by single nucleotide changes, and the other 230 require a double or a triple. And the deep mutational scanning experiments just cover all of these. So we have the nominal fitness distribution for those. We don't have a good mutation rate model, but we just know that they happen at a different rate, so we can treat those as the singles and the multiples as 2 different rate classes. And that's what's shown here. So in the nominal distribution, when we separate the multiples and the singles, there's a slightly higher selection coefficient for the multiples. Again, if we use this rank order statistic and we ask how likely is it for a multiple to be more fit than a single, it's about 60%, so it's slightly higher than 50%. Whereas in the fixed distribution, even though there's very few multinucleotide drivers, okay, because the rates are so small, they tend to have a considerably larger benefit. So here, using these rank order statistics again, the chance that a multinucleotide change is more beneficial than a single nucleotide change in the fixed distribution is 88%, and that's a significant difference. Okay, so I need to wrap up here. I just want to say this project's been going on for a few months. It's been very exciting. We have interesting theoretical and empirical results, but the analyses of actual distributions, we have to really interpret these with caution because we're using these data in ways that are potentially problematic, and I can talk about that in the questions if anyone wants to ask. It's been a delight to work with this group of collaborators. Brian Gitschlag from Cold Spring Harbor did the theoretical work on non-causal associations. Alejandro Cano from the Swiss Federal Research Institute in Zurich did the empirical analysis and the simulations. And David McCandlish and Josh McCain are co-supervising with me, and I'm looking forward to working with them on this and our other ongoing projects. Okay, so just to wrap up on the bigger picture, this year this theory of mutation-biased adaptation passed an important milestone. which is the final stage in the progression of truth. So in which a disruptive new idea is first there's rejected as absurd and disputed as being unlikely and dismissed as unimportant. And then finally the idea is claimed on behalf of tradition and attributed to dead authorities. And in evolutionary biology, the strongest display of sympathy for an idea is to attribute it to Charles Darwin. So in this recent commentary on, this was from a press release about Clonal, a paper from this spring that adds to the growing empirical case that mutation bias influences adaptive evolution, helping to return Darwin's second scenario to its rightful place in evolutionary theory. Again, with regard to the bigger picture, so this idea that mutation shapes adaptation has gone from being heretical to being controversial to now being established, although it's not like widely taught and considered. And so the question now for the field is not does this happen, it's just when does it happen, under what circumstances, and how much of it, and what are the implications of this. So today I gave you an example of a new implication that was a little bit surprising and counterintuitive that certainly changed my own thinking about how to interpret data. The way that this case for mutation-biased adaptation was established is detailed dissection of cases of adaptation, this very labor-intensive phenotype-first approach. But the way that we have to go forward with this is really with these high-throughput methods where we're characterizing entire distributions. And ideally getting precise and high-throughput analyses of the nominal distribution, the de novo distribution, and the fixed distribution. And finally, the big picture for evolutionary theory is that over the past 30 years, there's been a huge amount of intellectual work on kind of internalist ideas about evolvability and self-organization and so on, but it's really been missing this element of what's the underlying theory of population genetic causation. And so the theory that I presented today provides a way to rethink a lot of these issues. All right, and with that, I am just going to wrap up. I'll answer questions in the remaining time, and anyone who wants to contact me online, I'm easy to find. Thank you.